Good morning, everyone. As Darby mentioned, my name is Juhyun Kim from Division of Bioequivalence III. Today, I'd like to talk about common BE deficiency in ANDA for ER drug product. Before going into detail of my presentation, I have a short disclaimer in the bottom of this slide. For next 15-minute presentation, I will start with the common BE deficiency for ER drug product, followed by first cycle BE adequacy rate and review time. And then I will share one case study with my recommendation. For learning objectives for today, I'm going to summarize common first cycle BE deficiency in ANDA for ER drug product. And then we are going to learn from the case study on BE deficiency in ER formulation to better understand the common BE deficiency impacting first cycle approval for ER product. And then we are going to identify a strategy to prevent some of this deficiency. Due to limit of time and resources, it was not feasible for me to look at ANDA for all ER drug product. So I focus on the tablet formulation, and then I selected the 8 drug product, which listed in the bottom of this slide. And this for 8 drug product, 157 ANDA was received until end of year 2024, and 224 deficiency was identified during first cycle BE review. This 224 first cycle deficiency can be categorized for different area, as you can see in the this slide. And most commonly observed deficiency topic was dissolution, and followed by bioanalysis and PK and formulation. So in each category, multiple deficiency was identified. I'm listing common deficiency in order of frequency and omitted some less frequent one. 37 deficiency was related to the dissolution. Common issue was missing or inadequate multi-pH media dissolution and dissolution data for score tablet. For QC dissolution, usually no problem in submitting data, but the lacking of discriminatory ability of QC method was the main reason for deficiency. For ER drug product, alcohol dose dumping study is recommended. Some of this deficiency mainly due to partial submission of alcohol dose dumping or increase of alcohol drug release from the test compared to the reference drug. 17 deficiency was related to the pharmacokinetic. Some ANDA exclude subject without proper justification or document, and missing inadequate information on the failed pilot study, especially in study report, and incomplete SAS transporter file was the main theme for this deficiency. 35 deficiency were related to the bioanalysis. Missing analytical raw data and chromatogram and inadequate sample reanalysis was the main reason for this deficiency. And missing SOP and validation and also stability data was another deficiency for bioanalysis section. For formulation, exceeded excipient level and missing information on colorant, flavor, and shell was the common deficiency. So the 157 ANDA was solely based on the receive date, which is pre-GDF 1 to 3. And then I look at how many ANDA was considered adequate during the first cycle BE and review time. And review time was counted from the FDA receive date to the first complete BE review with the outcome of either BE adequate or inadequate through the CRL. And first cycle BE adequacy rate was significantly increased from the 11% to 83%, and review time reduced 22 to around 7 months as GDPA program matures. Case studies about dissolution testing using SCORE tablet for metoprolol-succinate ER tablet. RLD was designed as ER tablet comprised a multi-unit system containing metoprolol-succinate in multitude of the control release pellet, and each pellet acts as a separate drug delivery unit. So as you can see in the picture, the RLD contains multiple pellets, and each pellet can control the drug release. And a film coating is just for easier swallowing, not for functional coating. Therefore, this RLD is a SCORE tablet and can be divided per RLD labeling. So for metoprolol-succinate ER tablet for 100 and 200 milligram, PSG recommend in vivo fasting and fed-B study on 200 milligram strength. And additionally, compendial dissolution testing on whole tablet and half tablet is recommended. This is because of RLD, the formulation design, and multimedia dissolution testing and in vitro alcohol dose dumping test recommend in accordance of general guidance. And 100 milligram strength can be waived based on criteria set forth. This is the hypothetical case for metoprolol-succinate ER tablet based on in-house ANDAs. In this hypothetical case, they have two strengths, 100 milligram and 200 milligram strengths. B study conducted on 200 milligram strength on whole tablet and considered acceptable. And dissolution testing, which is alcohol dose dumping and multi-pH media study, were considered and for both strength. And two graphs in the slide are results from the compendial dissolution testing using whole and half tablet. For 200 milligram strength, RLD drug product, half and whole tablet show almost identical dissolution profile. On the other hand, for test drug product, half tablet show much faster drug release compared to the whole tablet, and same trend observed 100 milligram strength. As discussed in previous slide, reference product comprise a multi-unit system. There is no functional coating. So therefore, dissolution profile for half and whole tablet for both strengths are comparable. On the other hand, test drug product showed half tablet is much faster than whole tablet for both strengths. And we think it was related to the product design, test product designed to have drug-loaded matrix in the core and functional coating outside. So as you can imagine, when test product is splitted, the functional coating is going to be compromised. So therefore, additional study were recommended. Applicant was asked to submit fasting and fed study on 200 milligram strength and half tablet, and additionally comparative multi-pH dissolution testing and in vitro alcohol dose dumping testing on half tablet. The reason this study is recommended is because this application has 100 milligram strength too, and alcohol dose dumping and multi-pH media is important for the waiver purpose. Alternatively, it was asked to reformulate test product. As a response to first cycle B deficiency, the applicant conducted a fasting and fed B study on half tablet of 200 milligram strength. Both fasting and fat study on half tablet failed because 90% confidence interval of Cmax did not fall within the limit of 80 to 120 and failed in upper limit. So this supports our concern that different formulation design may have impact on bioequivalence of a half tablet. Therefore, applicant was asked to reformulate. Based on this result, we can learn that a good understanding of formulation design of RLD drug product would help to avoid deficiency and reduce the time and effort in drug development process. This slide summarizes my recommendation for avoiding BE deficiency. For dissolution study, please check the PSG and relevant general guidance for identify adequate dissolution testing. Please generate dissolution data using discriminating method. It will be helpful if you submit the method development report or supporting data to show discriminatory ability of your method. Please make sure the stability of test and reference standard product at the time of dissolution testing. For bioanalysis, please submit the 100% analytical numerical raw data, including reject and repeated run, not just for the accepted run, and chromatogram from 20% of CDER, not randomly selected subject. For sample reanalysis, please provide pre-specified reason and proper justification with the supporting document and data. For formulation, please provide quantitative breakdown of the colorant, ink, flavor, and capsule shell. If you need clarification on whether proposed level of inactive ingredient in the test product are acceptable, please submit the control correspondence. For PK, if you exclude the concentration data of subject in BE statistical analysis, please submit the investigation report with the real-time evidence for protocol violation. Please provide SAS data in proper format of data in SAS file, which match with the presented in the D study report. So all clinical trial studies starting after December 17, 2016 are required to have data submitted in CDISC compliant format, which lists in detailed information in this link. As a summary of my presentation, out of 157 ANDA, 224 deficiency were identified in the first cycle BE assessment for ER drug product, and various deficiency identified during BE review for oral ER product are avoidable. Submitting thorough, well-structured, and acceptable BE data for ANDA will greatly benefit generic drug industry and the American public by facilitating faster and wider availability of generic drug. For today's talk, I'd like to thank DB3 management, April Ki for support, and Lee Gong for initiating this project, and Fang Lu for helping feedback during clearance process, and OB management, Dr. Bing Lee and Partha Roy. Thank you for attention.